All right. Welcome back, everybody, to lecture 16 now, or lecture 2 for Professor Rahola Baker. Today's lecture is going to be more about epigenetics and all that stuff. So let's go ahead and get into it. Talked about a lot of this. Epigenetics can lead to cancer. You can imagine that if you use your environment to modify genes that maybe cause cancer, maybe it modifies a proto-oncogene so that it's more expressed, whatever, it could lead to cancer, stuff like that. So epigenetic they can change chromosome structure and affect expression and cell function, right? So not a surprise. So here is a let's see here. Yeah, so I think what they're trying to get at with this here is a HAT generates a positive transcription mark, meaning it promotes transcription. So if you lose the thing, is it going to cause cancer through tumor suppressor or oncogene? And I would say it's probably going to cause it through tumor suppressor here, because if this promotes transcription and you're getting rid of it, and you get rid of transcription of a tumor suppressor, you're not going to suppress the tumor anymore, and therefore it's gonna blow out of proportions and divide uncontrollably and stuff like that. So that would be my logic for that. Already talked about the nucleosome. Remember, it's made up of 8 histone cores. I can't draw all of them here because they're all like underneath, and then each of them have a tail, right? And then there's also an H1 that holds things together. So how does the cell modify nucleosomes? We talked about this. There's all sorts of different enzymes. So there's erasers, readers, and writers. There's also things like nucleosome remodeling complexes, which can change a full nucleosome into a half and half. It can just completely switch it out. It can do things like maybe changing quarters out of it. It can do a lot of things change. It can do a lot of things in terms of that. And then tail changing, right, is done through modifications. So the tail code is this is specifically done through erasers, so erasers and writers, right? And how does the cell recognize the histone through readers, right? Readers would be like I spelled that horribly. Readers. So it recognizes through readers, right? So here's an example of a protein, PHF8. Remember, it's both a reader and an eraser. So it reads H3K4ME3, and then it demethylates H3K9ME2. So it reads the ME3 and demethylates the K9ME2. So it's both a reader and a eraser. Here's a bit more on that, by the way. So it binds to the marked promoter and removes the repressive methyl marks to activate transcription. So it promotes transcription by binding to this, removing this repressive methylation or dimethylation, and promoting transcription. Already talked about this. So how is the histone tail code recognized? It's just going to be a active site thing, right? Or like there's going to be a binding site that's specific for that symbol, or for that amino acid modification, right? Specifically. Already talked about this a bit. There's more complex versions of it, right? It doesn't have to just be the simple ones that we've seen before. You can have many different readers attaching to a scaffold, and then that recruits a massive monstrosity or whatever, and that does other random things. That's what they're trying to get at here, right? So you see here, again, reader complex recognizes histone tails, binds to another complex, so this purple is supposed to be the complex with writers and erasers, and that causes downstream modifications. And that leads to biological activity. Okay. I wish I want them to just give, yeah, okay, so I want them to just give me all of them. Yes, okay, so this slide. Very important slide. These, you should remember these, put them on your note card, do something. So the a lot of these are just markers for how things interact. So, for example, for promoters that are bound by polymerase 2, so that'd be like RNA polymerase 2 if I'm not mistaken, or sorry, mRNA, or just any RNA, because right, yeah. So if it's any RNA polymerase 2 bound promoter is marked by this lysine. So basically what it means is that helps recruit the lysine, or sorry, that helps recruit the polymerase, if you want to think of it like that. So whenever this is present, the polymerase 2 is bound. So this is very helpful with binding a DNA polymerase 2. So that's important. That's what the K4 is for. The K7 is, or K27, sorry, is a broad repressive modification. So it occurs at promoters, gene bodies, and enhancer regions. And there's a couple of cool interactions that this has. So if you couple this with a K4ME3, it indicates a poised promoter. And a poised promoter, let me just get you a actual definition, is basically a promoter that is inactive, but it's like ready to turn on at any moment. So it's kind of like being repressed, but it's also ready to be active, if you will. And then co-localization with a separate one, which is H3K4ME1, indicates a poised enhancer, which is, again, an enhancer that is, like, ready to be active, but it's not quite active yet. So these are just some things that you should keep in mind. So this H3K27ME3 is a repressive marker, but when it's coupled with some of these activating markers, it leads to a promoter that is, like, ready to go and it's been revved up. The last one I'm missing is this H3K27 acetylation, or AC, is marked by promoters. Marks promoters, not is marked by. So this thing marks promoters that are active and actually working, and also marks enhancers that are actively working. And this specifically is read by bromodomains. You don't have to know the ins and outs of bromodomains, just know that they are sections of proteins that bind to specific modifications, and in this case it binds to this. So we've talked about a lot of different proteins and a lot of different modifications. So here we have 3 examples. We have some that induce transcription, like this guy here. We have some that repress it. We have KME3. And then we have, some that do it in the between, like when you co-localize them, right? When you add the K4 and the K27 together. So, yeah. So cis and trans actions, I'm going to be honest with you, I think they describe it a little bit better down here. Also, I'll talk about that a little bit later if it's present. If it's not, I'll come back to it. But cis and trans is like, does it... when it's bound to, I believe it's like when it's bound to a section of DNA, does it assist with transcription, translation, or does it modify expression of a gene close by or of a gene very far away, is what the idea is, I think. We've already talked about this. I'm not going to harp on this too much, but 3 examples of histone modifications. So H3K9ME3 is... not shown up here, is it? No, it's not. That's okay. But this is another modification, leads to... oh, yeah, okay. What they're trying to show you here is these different modifications. So you can see here, we have an H3K9ME3. This is resulting in a heterochromatin formation, so gene silencing, right? Because euchromatin's open, heterochromatin's closed, remember? So when you do this, it's another inhibitory or repressive modification. When you put both of them together, right, the K4 one, remember, is this Me3 guy. Remember, when you put this and this together, it leads to a poised promoter. That's kind of what's going on here, at least the gene expression. And then for the trimethyl, this is the K27. This is the one up here, right? That just leads to repression. Right? So many different types. Oh, perfect. They did define them. Okay, cool. Right. So cis and trans actions. So a cis action, right, is when the change in a histone tail has a direct effect on the DNA. So the example given here is actually a great one. Yeah. So lysine with a positively charged nitrogen can bind the negatively charged phosphates of the DNA backbone. Acetylation neutralizes that, loosening the histone DNA interactions, right? Yes. So it's, let's see here. I'm trying to think of a way of describing this really in a good way. Maybe create binding site for chromatin modification enzyme. Yeah. So yeah, I should take back what I said earlier. I probably should start speaking before I know something, but it's okay. So for the trans actions, or sorry, for the cis actions, right? You can think of it as one action leads to a result. Whereas for trans actions, you can think of it, one action leads to another action, which leads to a result. Right? So cis actions are immediate, and trans actions take a bit of time, or take extra steps. So for a cis action, right, we have a lysine. Lysines are positively charged, and so they will bind to the negative phosphate backbone of DNA. If I acetylate the lysines, then the lysines are now neutral, and the DNA uncoils from the proteins, or the histones, right? And so what that allows for is that loosening directly allows for transcription factors and proteins to interact with the DNA. That is a cis action because it's one modification to a histone tail that results in a change in expression for a gene. A trans action is a little bit different. A trans action is what happens is you do the same thing as a cis action, basically. So you modify a lysine, but that lysine, just because it's modified, doesn't actually result in a change in the DNA. But what it does do is that modified lysine now recruits a different enzyme, right? Like a reader, right? So it'll recruit a bromodomain or some enzyme. And then that bromodomain will maybe attract another enzyme, or maybe the bromodomain is like the PHF8 or PHF8, right? Where it has a reader and a eraser domain. So maybe it's both, right? So it's gonna you're gonna modify the histone tail, and then another enzyme is gonna kind of come in, and that enzyme will have an effect on the DNA. Maybe that enzyme will transcribe maybe that enzyme will like modify the DNA and change its chromatin state so it can allow transcription or something like that. So it's like a secondary effect for trans actions. So spreading histone tail marks, what would make us think that marks spread? This entire thing here is just a example of how the position of these markers actually affects gene expression and stuff like that. So all this is just talking about so ommatidia. All you have to know about them is that they're important in the function of the fly eye. You really should not have to know all of this, I don't think. I feel like it's a little bit excess. But what you should care about is this. So normally, right, normally in a Drosophila, gosh, so many names that I keep messing up in this class. Anyways, normally in a fly, right, you have a white gene in this position, and you have a section of heterochromatin over here, right? So this is very let's see here. I just realized this is really weirdly drawn. Huh. Yeah, this is really, really weirdly drawn. it should be described in a better way. I think what they're trying to show here is that this white gene actually represses the white gene. And so when you put it in the heterochromatin region, it represses the repressor, and so now you get white parts of the eye. So basically what's happening here is this gene in a normal chromosome is very active and present, while the heterochromatin region is just stuck, right? But there's a rare inversion that can occur in this mutant where the heterochromatin has a section that's kind of switched, or like a section of the DNA switches. So this part and this part switch. And so now you have the white gene now located in a section of heterochromatin or very close to it. And because of that, the white gene doesn't get expressed, and so therefore the white gene doesn't repress the formation of white ommatidia, and so you get a white fly eye. And here they talk about a lot of different things. All they're trying to say here is that the genes involved in these kind of movements of chromosomes and stuff like that are epigenetic regulators, is what they're trying to say, I believe. So I genuinely don't think that this stuff is that important, to be honest. There's a lot of stuff in these lectures that I don't think is super helpful. But there's also a lot that's super helpful as well. So I don't know, it's one of those things where you have to pick and choose what's important. So screens for mutations that suppress or enhance white variegation have identified many conserved epigenetic factors, including lysine, whatever. Yeah, right. So basically what this is saying is there are a lot of different factors that are epigenetic. In this case, the histone modifications, right? That are impacting whether or not the fly eyes are white or red or whatever, right? At the same time, there's also other systems in place. So like in this case, we have a HP1A. What this does is it actually binds to this K9, and it interacts with the this again, which again, you guys don't really know what this is. But what this is basically talking about, in a nutshell, is that there are some proteins that recognize a certain set of signals, and they spread those signals to other parts of the genome. That is what it's trying to say. I wish that was explained a bit better. Yeah, and this entire section here is just detailing that process more. Gosh, yeah, this is, there's a lot of stuff here. I really don't think you need to know all this. This is a lot. I think what I would say here... Yeah, so all this is trying to get at is the spreading of heterochromatin, and so what you should know is that there is this HP1A protein, right? This thing will find and bind to things like H3K9 ME3s or ME2s, and this HP1A recruits a bunch of things. It recruits... I wish it would tell me. Yeah, so see this, right? So this, you know what, the image is actually pretty good. So the H3K9, right, methyl 3, recruits this HP1. This HP1 then recruits this 420, and then this 420 trimethylates this 20, right? And so that methylation results in a heterochromatin propagation. So this is a reader-writer complex, right? This whole thing, because the HP1 reads this ME3 on the K9, recruits these things, which are writers, and then those writers add the trimethylation to the 20, the lysine 20, right? That's what it's trying to get at. And so the addition of this causes chromatin to self-propagate, or the heterochromatin to self-propagate, because the trimethylation invokes compression, if you will. Yeah. Yep. So you can see how they're talking about it here. So you see how it's like this initially started on H3, but it's being spread to the next one over. So that's what it means by it's self-propagating. I don't think this is very important. I already talked about this a lot, but so for this protein, so this interaction, what they're talking about here is let's see here. Complex of reader and writer proteins can spread specific chromatin modifications along chromosome. Oh, yeah. This is exactly what I talked about up here, pretty much, right? Where certain complexes can spread certain combinations of regulation. So if you see here, we start with, there's maybe some regulatory protein in our DNA, right? There's going to be some histone modifying protein that binds to this protein and then leads to a histone modification, whether that be a methylation, acetylation, or whatever. Then a reader protein comes in and binds to that modification, which recruits another writer, which leads to another modification. And this chain continues over and over again, right? And so you're basically just forming new reader and writer complexes. And the formation and addition of these new modifications results in compaction of the DNA. And so this regulatory protein kind of serves as a barrier, right? That's why you see like a barrier, like so barrier, right? So this barrier, right, kind of serves as the point where it stops, right? So anything after this point is euchromatin. That's what it's trying to get at here. Okay, so yeah, we can think about this question here. So in a human red blood cell, HS4 acts as a barrier between beta globin. Okay, so let's just draw this out. This is the last slide, I think, right? Yeah, so HS4 acts as a barrier between beta globin. So I'll just say HS4, and I'll say beta globin, and locus and condensed chromatin read. So this is going to be condensed, right? So I'm just going to call it C, or con, right? So defects in the barrier cause severe anemia. So this beta globin domain must be important for red blood cell production. And by destroying this barrier domain, what happens is that remember, the heterochromatin is self-propagating. So this condensed section spreads into the beta globin, and this now no longer gets transcribed and translated, and so you lose function of this gene, which clearly is important for red blood cell function. And so that's the kind of critical thinking you kind of need to be using on these kinds of questions, right? But hopefully that makes sense. That's why it would cause anemia, because we no longer have the function of this gene, and it seems like it's important for red blood cell production, right? So, yeah. I hope the video was helpful. I know I went quite fast, but I just want to try and make sure I'm getting the content you actually need down without like going over everything that is just in the lecture. So, especially for this half of the quarter. So, yeah, I hope the lecture video was helpful. If you have any questions, leave them in the comments. Feel free to rewatch for Hola Baker stuff as well if I didn't explain something super well. But yeah, other than that, best of luck and happy studying.